words. Can you hear me? Can you see my screen? And do you just see one screen right now?
Yes.
You don't see the next slide, correct?
Yes.
Okay. Thanks, guys. You can tell I'm a real pro at Zoom, right? I'm technologically advanced. So the title of my talk today is Targeted Molecular Panels for Resistance Detection. And I thought I'd talk about it because, yeah, I'm interested in antimicrobial resistance, but antimicrobial resistance is going in a new direction, or at least in part, and that's kind of genotypic detection of antimicrobial resistance. And I thought it might be fun and hopefully useful to review what's out there in this space. But first of all, I absolutely have to celebrate all of you on the Zoom call. Happy Medical Laboratory Professionals Week, as some of you stated earlier. This is probably one of the most difficult times that we as a community have ever addressed as a clinical microbiology community, and we're doing amazingly. We are stepping up, and we are helping people throughout the world, just doing what we do every single day. To that end, I'd like to just, at the beginning of the talk, acknowledge the lab that I work with. This is just a snapshot of some of the folks in the Weill Cornell lab, and they are incredible people. I don't tell them how good they are enough, but they're amazing, and they've been particularly amazing during this horrid year. I do have to tell a lab week joke. I always like to do lab week jokes. And they're horrible jokes. Well, you tell me afterwards. What you get if you eat salmon and Nutella? And this is this was a joke told to me by my lab manager a couple of years ago, and I loved it, but I always remember it. What do you get?
I'll be stumped.
You're stumped?
Yeah.
You get salmonella. If you eat salmon and Nutella, you get salmonella. Terrible joke. I'll never do another joke again. Anyway, here are my financial disclosures. Good thing about Zoom is that you can just shut someone off, right? If they are too terrible. If I'm in person, it'd be a little bit harder to do that. So here's the objective. A single objective for this talk is to describe the advantages and disadvantages of molecular resistance panels and their optimal implementation in the clinical microbiology laboratory. So just provide a little introduction. What's the importance of antimicrobials and the importance of antimicrobial resistance? Well, this is, to me, a profound statement. Antimicrobials are unique among all therapeutic agents in that the use of the agent in one patient can compromise its efficacy in another. That is, resistance to that agent can arise. Again, we're no stranger to this statement. In the United States, each year, close to 3 million people acquire an antimicrobial resistant infection, and consequently, more than 35,000 people will die. And I always like to state, don't like to, but I think I have to state that we have entered a post-antimicrobial era where conventional antimicrobial therapy is ineffective in a significant number of cases. And this is despite also the development of novel antimicrobial agents. I can say that during COVID, when we had some of our ventilator-associated infections, patients who'd been on ventilators for 30, 40, 60 days, we were growing some really nasty organisms. Acinetobacter baumannii isolates that were NDM positive, that were resistant to ceftriaxone. There really wasn't much to treat them with. I almost shudder kind of thinking about what the physicians were doing to treat these nasty bugs. But you can see how one horrible situation can be a setup for antimicrobial resistance. So into the meat of the talk, what's the goal of these targeted molecular resistance panels? So before we should think about what's the goal of conventional susceptibility testing. So traditionally we do phenotypic susceptibility testing. My personal favorite is disk diffusion. We generate zone of growth inhibition sizes, we measure them, we interpret them, and we generate an antibiogram, and we release this information to our physician colleagues. Well, with genotypic antimicrobial susceptibility testing, we have an organism, and this is my stylized image of a Pseudomonas aeruginosa isolate. You'll see it in MoMA soon, I'm sure of it. And this Pseudomonas aeruginosa isolate has DNA in the form of a chromosome or plasmids. And that DNA, that genomic content, can be interrogated to understand if there are antimicrobial resistance genes present, or if there are mutations in bacterial genes that can give rise to antimicrobial resistance. We can use a number of different methods that I'll talk about in a second to do this. And just by detecting the presence of a gene, we can infer resistance. Detecting the presence of a mutation, such as one of those associated with fluoroquinolone resistance, we can infer resistance. We can also infer susceptibility as well. So if we don't detect the gene or we don't detect the mutation, we can infer that the isolate, whether isolated in culture, recovered in culture, or present in a clinical specimen, like a positive blood culture, is resistant or susceptible. And we can generate an antibiogram, much like we can with conventional susceptibility testing. I think it's really important, though, to state that antimicrobial gene detection or mutation detection only supports susceptible resistant interpretations. There's no intermediate category here. What's the challenge? Again, here's our wonderful, well, maybe it's not wonderful, Pseudomonas aeruginosa isolate. And antimicrobials have a very diverse mechanism of action. We have our antimetabolites, the targets of the sulfonamides and trimethoprim. We have the quinolones, which target DNA gyrase and topoisomerase. We have RNA polymerase, which is targeted by the rifamycins, also targeted by fidaxomicin, which is actually a macrolide in terms of its chemical structure. We also have inhibitors of protein synthesis, the aminoglycosides, glycosamides, macrolides, and the oxazidolones. We have the famous cell wall synthesis inhibitors, the mighty beta-lactams, which inhibit penicillin binding proteins or transpeptidases, and also fosfomycin that inhibits mu-A. And then we have the rather nasty polymyxins, but effective when you really need them, although they're no longer really recommended for serious carbapenem-resistant Enterobacterales infections that disrupt the membrane and cause leakage. So what I hope I've shown you here, that in addition to having a diverse mechanism of action, you can imagine that bacteria, in order to combat these wide array of antimicrobials, also have to have diverse antimicrobial resistance mechanisms. So how do you build in tests? What's the complete test for detecting all these antimicrobial resistance mechanisms at the gene or the mutation level? And this is something that's really important. Absence of an antimicrobial resistance determinant does not necessarily imply susceptibility. And I think this is really true of the gram-negatives, particularly the Enterobacters and some of the Klebsiellas and the Acinetobacters and the Pseudomonads. Really the Enterobacters, the Pseudomonads, and the Acinetobacters. Absence of an AMR determinant does not imply susceptibility. We can infer resistance by detecting the marker, but we can't necessarily always infer susceptibility. And that's because some antimicrobial mechanisms are inducible, and you might be looking at upregulating of proteins that are typically in a kind of silent state, and they get de-repressed, and then you have full-blown resistance. So what are the methodologies, the methods that we use for detecting resistance genes or mutations? Well, the classic is the wonderful PCR, where we have a target DNA, which is shown here, and my kind of stylized imaging again is our DNA molecule. And then we add in a cocktail of DNA polymerase and nucleic nucleotides and some primers to initiate replication. And we, in a cyclic manner, can amplify that original target molecule into billions of copies of that target molecule. And we can ultimately generate enough copies that we can detect the DNA molecule that was in that original specimen. There are also hybridization microarray methods where we have a solid surface, and on that solid surface are immobilized these small little oligonucleotides, which are complementary to the target molecule DNA molecule that we're trying to detect. Again, here's our little DNA molecule here that we want to detect. Let's call it MEK-A, one of my favorite resistance determinants. Then there's the next step where that target DNA molecule hybridizes to that complementary, those little complementary probes. And then we need to detect that hybridization complex in some way. And we can do that by using probes that also bind to a different region of our target molecule that has some type of structural or chemical detection methodology associated with them. One instance to have these kind of gold beads that light up. And you can actually see on the solid surface, if you can see those gold beads in a way through kind of microscopy-based methods, and you can infer that you've detected your given DNA molecule using a hybridization microwave method. I will say, guys, if I say anything that's stupid or I move a little bit quickly, please do shout out. This is the wonderful thing about Zoom, is that we can do this. So I'm going to keep looking at my chat box or my question box, and if you see a question come in and I don't notice it, please, Mary Claire, yell at me and I'll stop and try and answer it. So what are the flavors of targeted molecular resistance panels? So I want to stress that again, and I think I did at the start, but I'll stress it again, that these panels can be used either with isolates that have been recovered directly in culture, or detecting resistance determinants in bacteria that are in a clinical specimen, such as a positive blood culture broth or a BAL specimen. So I've kind of broken down these panels into 3 different groups, and maybe you agree or don't agree, but it's how my brain thinks about them. There are panels that I kind of call the kind of simpler panels that detect 1 or 2 organisms and all resistance determinants. Examples of that are nicely provided by Expert, the Expert system, manufactured by Cepheid, the Expert MRSA, Staph aureus blood culture system, the Expert VAN-A, which detects the VAN-A gene in enterococci. Then we have the so-called syndromic panels, kind of medium-level panels that detect microorganisms and their associated resistance determinants pretty much exclusively in clinical specimens. We have the film-array BCID panels associated with positive blood culture broths. We have the pneumonia panel that is associated with the Univero LRT BAL panel. So detecting resistance markers in organisms in BAL specimens, respiratory specimens. Then we have panels that really focus. I should say these syndromic panels don't have a huge number of molecular markers, typically about 5. And so this allows me to kind of differentiate into the last group of panels where we detect umpteens of molecular markers. So panels that can detect typically more than 5 resistance determinants, equal to all 5. And an example of that is the Expert CARBA, which just detects the 5 major carbapenemase genes that we see, that we typically see. And then there's these kind of giant panels that I'll talk about a little bit later, the Acuitas AMR gene panel that detects a whole number, like 47 different resistance markers. So I just wanted to break out some of these panels a little bit and discuss about them. I think the first one that kind of really launched onto the market in terms of how it was used for managing infections, when we really think about the resistance marker. So we've had the ability to detect organisms in specimens before, but detecting the organism and the resistance marker, it's relatively novel, well, within the last 10 years or so. And only now we're starting to see outcome studies that are really addressing the power of using these panels. And I'll talk about that in a little bit later too. So the first one I want to touch upon is the film-array BCID panel, which has 3 resistance markers on there. For the gram-negatives, Acinetobacters, Enterobacterales, and Pseudomonas aeruginosa have the potential of detecting the gene that encodes the KPC carbapenemase. So by virtue of detecting this gene, you would infer that the organism is resistant to pretty much all beta-lactams. I will say that in our lab, we don't actually report it out, the KPC gene, if detected. If it's not detected, sorry, we don't report it out if it's not detected if we detect Acinetobacter or Pseudomonas, because in our report, an individual might infer, oh, Acinetobacter species detected, KPC negative. And they might be thinking, oh, that means that the Acinetobacter isn't likely to be resistant to carbapenems or beta-lactams. The reality is that Acinetobacter and Pseudomonas are typically resistant to carbapenems by other mechanisms, not KPC. KPC is really primarily associated with the Enterobacterales. But things always change and there are exotic things. And a couple of weeks ago, I just came across a Pseudomonas aeruginosa that was KPC positive, and I kind of fell out of my seat, but it was real. So I just kind of contradicted myself. And then on this panel, we have gram-positives markers for MEC-A, which we'll talk about a little bit later, MEC-A and MEC-C, for detecting methicillin or beta-lactam resistance in Staphylococcus species. And then we had the VAN-A and VAN-B genes, which confer resistance to vancomycin in Enterococcus species, and notably Fecalus and Faecium. So a little bit about the challenge with the AMR panel, and I'm going to talk about the challenges as I kind of move through. Something that kind of burnt us a little bit, and it's unfair to say burnt us, but it was a learning experience, is that there are real challenges when you are using molecular panels to detect organisms and resistance determinants directly from clinical specimens. There's a real challenge in assigning which organism you detect actually harbors the resistance determinant, and it has consequence. And the example that I love to show is this example here, where we have a biofire report, and we've clearly detected MEC-A, the gene that confers beta-lactam resistance in Staphylococci. We've clearly detected Staphylococcus, great. And we've gone a little bit further and we've detected Staphylococcus aureus. So by all accounts and purposes, I'm thinking we've got MRSA here. I'm inferring that. Well, you subculture out the plate, not my streaking, by the way, someone else's. You know, mine's much tidier than that, teasing. But here, streaking out the blood culture, and this was directly from the blood culture broth on chocolate agar. I'm seeing that wonderful yellow Staph aureus. Then I'm seeing these tiny little colonies that don't quite look like Staph aureus. When you go back and you interrogate the isolates themselves, what you've actually got here is methicillin susceptible Staph aureus and methicillin-resistant Staph epidermidis. This being a single set, as it was, for me, the pathogen is MSSA, and the methicillin-resistant Staph epidermidis I don't really care about. The physician could have used an anti-staphylococcal penicillin or cefazolin, but instead, based on that initial report that we provided, went straight to vancomycin. So manufacturers have tried to get around this, and notably the Cepheid group did this a number of years ago, and recently the BioFire group have developed their panel, the BCID panel, to allow one to assign carriage of MEC-A to Staph aureus, and that's a feature that's present in the BCID panel, BCID2 panel, and here's how it works. So to be methicillin-resistant Staph aureus, you have what's called the staphylococcal cassette chromosome, and it's this element that kind of inserts itself into a specific junction in the Staph genome. I try to show it here schematically. When this DNA inserts into the Staph aureus genome, it creates this unique junction. Let's just call it MREJ. If we design primers that will detect the gene that confers resistance to beta-lactams, the MEC gene, if we detect that junction, and if we detect the Staph aureus chromosome using primers, I think we can accurately say what we're dealing here with is MRSA. So this is a Staph aureus isolate that has MEC-A inserted into the chromosome of Staph aureus. And as I alluded to, there's similar functionality associated with other systems, such as the expert system. Again, I'm just moving through these panels. This is the BCID2 panel, which is the kind of upgrade from BCID. And you can see, in addition to the 3 resistance markers here, are a number of other markers, pretty much exclusively associated with detecting resistance in gram-negatives. A number of resistance markers for detecting resistance to cephalosporins, the CTXM here, and a number of carbopentamases, and also rather intriguingly, the inclusion of a marker of primers to detect Mary Claire's favorite protein, MCR1, which confers resistance to the polymyxins. So BioFire aren't the only people on the block. There is also the Verigene system, which is broken up into 2 different panels, one for gram-negatives, detecting this suite of resistance markers, and also the Verigene gram-positive panel, which detects very similar markers to the BioFire panel. And then finally, ePlex has also 2 markers, GemMark, which I think is now Roche. I'm not sure, everything changes these days. ePlex has 2 panels, the gram-negative and the gram-positive, with very similar markers to those I've already talked about. Very interestingly, though, I did like this one. I suppose it's not too practical in the United States. All of these panels have the ability to detect MEK-A, which is the major, encodes the major PBP2A protein that we always think about with Staph aureus. There's the alternative PBP protein called PBP2C that's primarily found in Europe that is covered by the MEK-C primers. Most of the other panels don't actually differentiate between the two, but the ePlex system actually makes the distinction whether it's MEK-A or MEK-C. Clinically, it doesn't make a difference. So let's talk about the utility of detecting just a simple resistance marker. So the one that I love to talk about is the detection of MEK-A or PBP2A, which is the protein that encodes that transpeptidase that's immune to beta-lactams, except for ceftaroline. So therefore, Staph aureus encodes MEK-A. It's completely resistant to beta-lactams, except for ceftaroline. So MEK-A encodes the penicillin-binding protein 2A, confers beta-lactam resistance in staphylococci. And so we can infer, and this is what we do, particularly from positive blood culture broths, the absence of MEK-A, or in a colony, we can use immunoassays to detect PBP2A. If we do not detect either of those, we infer that this is MSSA, and we can treat with an anti-staphylococcal penicillin or cefazolin. If we detect MEK-A or PBP2A, we infer we have MRSA, and we do not treat with a therapy for MSSA, pretty much vancomycin or daptomycin. There are numerous assays cleared for detecting MRSA or MSSA in a range of clinical specimens, typically with very fast turnaround times. There are also 2 FDA-cleared immunoassays for detecting the presence of PBP2A in Staph aureus recovered in culture. Turnaround time is 5 to 20 minutes. So what's the clinical impact of these types of assays? And this is something I become increasingly interested in because, and as I know that many of you on the Zoom are, we traditionally as a community didn't cost much, right? We were not really viewed as a high spending part of the hospital. It's the automated analyzers and chemistry. But then we started to be a little bit more comfortable asking for technology that was going to revolutionize what we did, mass spectrometry, syndromic panels. And then people started asking, well, okay, this is quite a bit of ask here. How do you know that this is actually working? So I really believe that as a community, we have to show that what we're doing works, and it does work. And we have to convince our friends to stop thinking this as just a little silo that only contributes to micro. We control everything. Microbiology touches every single part of the hospital and every single part of the community. We need data like this to show our bean counters that we are making a difference. So this is 2 stories that describe the clinical impact of detecting MEK-A in Staph aureus. One is a pre and post intervention retrospective study. Put simply, they had a pre-intervention period where they had 106 subjects that were treated with standard of care therapy, or standard of care clinical microbiology diagnostics. Then they had a post-intervention group of 120 people, where in this post-intervention group, they had specifically implemented a molecular assay for detecting Staph aureus and MEK-A, if it was there, in positive blood culture broths. If Staph aureus was or was not detected, this information was relayed to an antimicrobial stewardship team 24/7. So what good did this do? Did a lot, actually. They found that they were able to, the time to initiate a targeted antimicrobial decreased by almost 21 hours. That's remarkable. The time to getting a targeted antimicrobial specifically for MSSA, so that anti-staphylococcal penicillin or cefazolin, decreased by almost 41 hours, which is redunculous when you think about it. As a result, and this is where we really, really want to show our power and our strength, is that this intervention was associated with lower in-hospital and 30-day mortality. So this is remarkable, in my opinion. And these are the data that we need to be sharing with our friends in the C-suite. Then another really lovely study. I'm always partial to it because the senior author is a wonderful ID doc at Columbia who loves clinical microbiology and is a sequencing guru. She really appreciates what we do in the lab, so I always like to celebrate her work. So this is a retrospective observational cohort study. And again, to put simply, between the periods of 2007 and 2015, they analyzed subjects or patients that experienced Staph aureus bacteremia. They broke it down into 3 different groups based on time, a group 1, a group 2, and a group 3. Just by random, as you might imagine, these technologies were blowing up. The clinical lab that was associated with Anchor-Trin's hospital implemented a molecular assay for detecting MEK-A and Staph aureus directly from positive blood culture broths. If Staph aureus, MEK-A was or was not detected, so I should say, clarify for both studies, if Staph aureus was detected, that information was relayed to the physicians. And the further information about whether it was MEK-A negative or positive was also relayed. And in this instance, in this study, an infectious diseases physician was called if Staph aureus was detected and whether MEK-A was or was not detected. So what did they show? So looking at those 3 different groups, you can see that there was a decrease in group 3 compared to groups 1 and 2 to the average time of initiating an anti-staphylococcal therapy to treat MSSA. What did that relate into? Basically, it related to a statistically significant decrease in MSSA mortality in group 3, so that group where that intervention of using the technology was in place, compared to groups 1 and 2. I think these are really powerful data, and I love to share them with people because I think it helps us get the technology that we need and we deserve. If COVID won't do it, this will do it too. This will get us the recognition we need. So what about detecting PBP2A directly in cultures of Staph aureus? Believe it or not too much has been done, and I think it's low-hanging fruit. But just to describe, molecular targets are not always just DNA. There are also proteins, and PBP2A can be detected using those immunoassays. There are 2 flavors. There's a latex agglutination assay that's actually FDA approved for Staph aureus and for coagulase negative staphylococci, or as Romney Humphreys likes to call them, Staphylococcus other than Staph aureus, because really there's lots of different flavors of coagulase positive staph circulating. Primarily they're animal pathogens. They're my other love in life. And there's also a lateral flow based assay that will detect PBP2A. Both of them are good. I have a little penchant for the lateral flow based assay because it's quick and it's easy to use, but its limitation is that it's not FDA approved for coagulase negative staph, only for Staph aureus. The one study that I'm aware of, and please, if I'm wrong, correct me, folks, there's one study that looked at the impact of detecting PBP2A directly in Staph aureus isolated in culture, essentially that they found that they were able to make a number of very quick interventions. So remember, this data would be available at least 24 hours before conventional susceptibility testing data would be available. So it's still very powerful to use these immunoassays. And the interventions that were involved, essentially switching patients from inappropriate to appropriate MRSA therapy, or optimizing targeted MSSA or MRSA therapy. What about the clinical impact of detecting carbapenemase genes in Enterobacterales? Remember, I talked about how a number of those panels actually have markers for KPC. Remember, that's probably the most common carbapenemase in the United States. So this is a nice study. I'm biased because I'm involved, but this is a nice study that is centered in the New York, New Jersey area. And I will say that this part of the world can be considered endemic for KPC producing organisms. And so it's an observational study of patients with carbapenem-resistant Enterobacterales bacteremia performed at 8 medical centers. And the objective was to determine whether use of either of the following was associated with a decrease in 30-day mortality. Again, so really looking at outcomes. And what was the use of the following? Either a molecular resistance panel that detects BLAR KPC on positive blood culture broths. So was using that panel that detects KPC associated with a decrease in 30-day mortality? And this was conducted at the time really when these novel agents, beta-lactam agents, were coming on the market that had activity against KPC-producing organisms were arriving. And so the study was also designed to understand if use of ceftazidime-avibactam, which will target KPC-producing organisms, was associated with a decrease in 30-day mortality compared to the kind of nasty, toxic polymyxins and the aminoglycoside combination therapy. So multiplex PCR was applied to a number of cases. So each case was an independent subject. Notably, the PCR assay in question was only used at 3 of 8 medical centers. And a fair number of the cases were actually positive for BLAR-KPC. I will say that epidemiology has kind of changed. We've seen a decrease in KPC-producing organisms from positive blood culture broths, which is only a good thing. So a lot of information here, but just want to draw your eyes to the yellow box. So this is what I think is really cool. Detection of BLAR-KPC using this molecular panel, the BCID panel, directly on positive blood culture broths, was ultimately associated with a decrease in the time to identification of carbapenem-resistant enterobacterales and a decrease in the time to using active therapy against that carbapenem-resistant enterobacterales. And ultimately, and this is what I think is really cool, a decrease in 14 and 30-day mortality. It was also shown that using that targeted therapy, so if KPC was detected on that molecular panel, the physicians used ceftazidime-avibactam as opposed to polymyxin aminoglycoside. That intervention was also associated with decreased 30-day mortality. I know I'm biased, and forgive me for being biased, but I think these are really powerful data that we should try and share with our greater colleagues, so they can see the good that we do as a service. When I say we, I don't mean me, I mean all of us. So what are the other panels that we can think about? There's a number of kind of launched onto the market for detecting pathogens in respiratory specimens. Notably, there's the film-array pneumonia panel, which is FDA approved for a number of different respiratory specimens, lower respiratory tract specimens. It detects a number of different resistance markers. Again, these are all associated with the following gram-negatives. Again, CTXM, which is a marker for extended spectrum beta-lactamase production, and then all these other genes which are associated with carbapenemase detection. And then we have our mighty MEK-A detection system with that ability to say whether the MEK-A is associated with Staph aureus to get at that coag-negative Staph aureus issue. They're not the only people on the block. There are also some wonderful systems from Caritas, which are cleared for these specimens, these organisms for gram-negatives, these markers. A little bit more when it came to detecting gram-negative resistance in the Caritas-based system, the TEM, the beta-lactamase TEM. Again, for gram-positives, detecting MEK-A. So what about these panels? How useful have they been? Well, again, folks, correct me if I'm wrong, but these are relatively new methodologies, and so not much has been done, but what has been done has been, in my opinion, rather good. And there was a kind of comparative study that was kind of headed up by Blake Buchan up in Milwaukee. And I think this is a really, really interesting read. So they applied this pneumonia panel. For disclosure, it was the BioFire panel, which can detect 33 different targets, which is kind of amazing when you think about it. A number of bacterial and viral, and also antimicrobial resistance targets, the ones that I just showed you earlier. It was a single center study, which is great. Sure, people love multi-center studies, but single center studies are a good place to start. 259 adult inpatients, and they exclusively assayed BAL specimens. And notably about the pneumonia panel, they only report an antimicrobial resistance marker if a target, an AMR target, is detected in the presence of an organism that makes sense. So if KPC was detected, and so was Staph aureus, the KPC result would be masked by the system, because KPC by itself with Staph aureus doesn't make sense because it's a gram-negative marker. I should say this is true for pretty much all syndromic panels, including the blood culture identification panel. They found, interestingly, that detection of MRSA using the pneumonia panel and using the markers that I've shown here, only had a sensitivity of about 82% and a specificity of 86%, which isn't great, honestly, when you think about what this means in terms of therapy. Again, only 18 cases though, so I don't want to really read into this too much. What I thought was kind of profound, if I can use that word, is that when they looked at the detection of carbapenemase genes, remember those 5 genes I talked about, including KPC, and also the ESBL gene, that CTXM gene, they were only detected in 4 of 53 specimens. And that's fine, that could be their prevalence. But I'm just showing you the table here of their results. And I just want you to focus on the panel markers here. KPC, so in 2 instances, so there were 3 instances of detecting, of those 4 instances, there were 3 instances where it was a carbapenemase gene, and 1 instance when it was an ESBL gene. You can see that there were some situations where there was discrepancy between what was detected. For instance, Enterobacter cloacae was detected by the panel, an NDM gene was detected, so a metallobeta-lactamase, which is kind of fearsome, but the isolate recovered was actually carbapenem susceptible, so there's a discrepancy. So when they looked at everything that in these examples, they found that they only had a 20% concordance between the AMR target identification and the phenotypic susceptibility of gram-negative isolates recovered in culture. Kind of depressing, if I can use that. It's a kind of strong word, but again, it's a limited study in an area that perhaps isn't endemic for multi-drug resistance. And I'd love to see a multi-center study looking at the performance of this panel to understand if there are differences in geography and differences in patient population that could see different outcomes. But what the authors also did, which I thought was very cool, they did what I call, and maybe it's wrong to call it this, a simulated antimicrobial stewardship study, where they looked at the potential impacts. So again, this is all done behind the scenes. This assay was not being used at the time for affecting patient care. So the behind the scenes, they wondered whether, if the physician had had the pneumonia panel results, could they have made changes to the patient's treatment, their therapy, that could have led to favorable antimicrobial stewardship endeavors. Okay, so they reviewed a number of patient medical records, including the clinically prescribed antibiotics, and they revealed that there was a potential for changing the antibiotic based just on the pneumonia panel in a simulated way in 71% of the instances. That's a lot of room for making an intervention at least 24 to 48 hours before conventional susceptibility data is available. They found the most common intervention that they could have made, again, it's all simulated, it's all potential, was an appropriate antibiotic de-escalation and discontinuation. And this makes all our ID docs and our antimicrobial stewardship pharmacists so happy. The crux was that they were able to really reduce the number of unnecessary antibiotic therapy hours, kind of look at it, how many hours a patient would be collectively, or the group of patients collectively would be on antimicrobials. They were able to reduce that duration by 18,000 hours in 122 patients. That's crazy. Think about how much reduction in antimicrobial therapy that is. They found that if they could have used this panel, they could have reduced the number of total days a patient was on an antimicrobial by about 6.2. And they found the biggest escalation, if we look at this graph here, was, de-escalation, discontinuation, was the ability to de-escalate or to remove vancomycin treatment and also piperacillin tazobactam treatment. They found, there's the converse, right? They found that the appropriate escalation or initiation of therapy, so that is, you detect something, so you big up what their therapy is going to be, was only affected with the pneumonia panel in about 4.3% of patients. But again, when they look at the data together, this would have allowed these patients to get on effective therapy 185 hours before, when just relying on conventional culture-based information. So again, I think this is really powerful, even though it's simulated behind the scenes. These are the data that we need to tease us so that we can go out and we can do these larger studies that show how good we are. So I want to tell you a little story about something that we did here. We implemented a molecular panel for detecting the kind of big 5 carbapenemase genes in isolates recovered in culture. It's the CARBA-R assay, and it detects these 5 guys here. Notably, when we think about carbapenemases, we think of them being metallobeta-lactamases, which are kind of a little bit more fearsome than the conventional serine-based carbapenemases. That's because the metallobeta-lactamases are usually much more difficult to treat. They're often immune to some of these novel agents that are coming out. So we have the IMP enzyme and the NDM and the VIM enzyme, which are metallobeta-lactamases, and the KPC and the OXA48 are serine carbapenemases. So this assay is FDA cleared for Enterobacterales isolates, also Acinetobacter and Pseudomonas, and also rectal swabs. But we just wanted to study Enterobacterales isolates recovered in culture. This was kind of a New York City experience where I told you we're kind of endemic for carbapenem-resistant Enterobacterales. And we analyzed meropenem non-susceptible Enterobacterales isolates that were recovered in culture. And so these were either intermediate or resistant. And we paged an infectious diseases fellow or stewardship pharmacist between 8 a.m. and 10 p.m. You can tell that the ID, you know what type of ID fellow you're dealing with, because sometimes you page him at 10:15 and like, dude, I'm off the clock at 10 o'clock. Okay, hands up. Sorry, I forgot you were a doc. I'm teasing. Well, not really. Sometimes you get that. And basically what we did, based on the result, the CARBA-R result, we would define a therapy, or the ID doc could define a therapy for the primary team to treat the organism that was recovered in culture. And again, this is another point I'm going to make, and I'm going to make here as well as later. It's really important, if you're going to use these panels, the best way to affect favorable outcomes is to have a guide, an easy read-to guide for your docs. So basically, we had a guide that was in the EMR, or rather a link to it was in the EMR. So you had to click the link and then it will come up, but you would click the link and it would say what gene was detected, and that would be in the patient's chart. Okay, KPC was detected. That's a class A enzyme, so that's a serine-based enzyme. Use ceftazidime avibactam, that's preferred, or polymyxin B, high-dose extended infusion meropenem. That's likely going to change based on the recent IDSA guidelines. Well, let's look at a metallobeta-lactamase, such as NDM. So we know that's a class B, so it's a metallobeta-lactamase. Our recommendation was polymyxin B plus a second agent based on susceptibility, or, and this is really probably the better therapy, is to mix up aztreonam and ceftazidime avibactam. If you recall, the metallobeta-lactamases are inhibited by aztreonam, which is kind of surprising, but these organisms typically have other beta-lactamases that will chew up the aztreonam. So you add in the ceftaz avibactam, the avibactam kills all the other beta-lactamases that would chew up aztreonam, leaving the aztreonam to kill the carbapenemase. It's kind of a cool two-pronged approach. So this was our therapeutic guidelines based on the results that we got from this panel. And the objectives of the study were to determine, just truthfully, the molecular epidemiology of CRE in this single hospital. And then we wanted to understand how the assay informed therapies and infection prevention and control investigations. So truthfully, this was a huge kind of interdisciplinary collaboration between stewardship, ClinMicro, ID, and infection prevention and control. So we had 135 CARBA-R tests that were performed during this time period. We excluded 84. It seems like a lot, but we excluded a lot because they were tested. 44 were tested on non-Enterobacterales. Got very excited when this assay came in, and I started trying to test all manner of Pseudomonas and Acinetobacter, and then kept reminding myself that those carbapenemases that this assay detects really isn't found in those organisms. But as I said, I did find that one KPC producing Pseudomonas aeruginosa, so maybe it was worth it. $3,000 later, it was worth it. And then we also excluded a number of other isolates based on the reasons I'm putting here. We ended up with 51 CARBA-R tests from 50 patients, and here's the breakout here. By far and away, the most common enzyme that we detected was BLA-KPC. Then we had an IMP, an NDM, a couple of OXA48s, and a VIM, and a number of them were CARBA-R negative, probably resistant to carbapenems by an upregulated cephalosporinase with a porin mutation, or more excitingly for me, maybe a novel carbapenemase that isn't detected by this panel. I haven't got around to looking at them yet, but I will functionally try and characterize them and also sequence them as well. So again, just focusing your attention on this yellow box. Most common analyte detected was BLA-KPC. Very interestingly, Of those 8 non-KPC enzymes that we did detect, over 50% of them were recovered from patients that had international travel within a month of presenting to the hospital. Montenegro, Bangladesh, and I think France for the OXA48s. Remember, France is, I think, the most predominant carbapenemase in France right now is the OXA48 enzymes. Antimicrobial changes were made in about 20% of the patients. Ceftazidime-avibactam was implemented due to BLA KPC detection. And as I said, that kind of cool trick of using aztreonam and ceftazidime-avibactam was implemented due to detection of NDM. Really interestingly, none of them were on active therapy prior to this result, the CARBAR result. And the median time on an ineffective therapy was 48 hours. I did spell check. God, I missed it. Oh, sorry. The median time to getting that active therapy after the CARBAR result was 3 hours. And we also used the system to kind of understand if there were clusters of infection with non-KPC enzymes. And so, for instance, the NDM producing patient was in a room with 3 other patients. We took rectal swabs for them, and we used the CARBAR to double check on the isolates that were recovered from the rectal swab if they were NDM positive, and they were negative. You know, what's really important in ClinMicro, as I hope I keep stressing, is that not only do we have to show our outcomes outside the lab, but we should also justify our costs in the lab. And I would say that we calculated that the cost to detect a single KPC enzyme was $110, which is peanuts when you consider about everything that this was doing. The cost to detect the non-KPC enzyme was about $400, which again is peanuts when you can consider how powerful these assays can be. In-hospital mortality rate wasn't really significantly different between groups, so I can't really say too much about that, but I will say there was no in-hospital mortality for the non-KPC carbapenemase producing CRE isolates, so the NDMs, the VIMs, and the IMPs. And just to remind you just how powerful these assays are and how they can direct therapy, when we look at standard susceptibility profiles to beta-lactams and these carbapenemases, again, we got our class A, the serine, got a metallo example, such as VIM or NDM. Then we've got a kind of another serine enzyme, but a little bit different from the class A, the class Ds, and the representative example is OXA48. You can see that when we look at our traditional therapies, the beta-lactams, the landscape is rather bleak, really very little susceptibility. As I said, for the class B enzymes, they're typically susceptible to aztreonam, but the isolates typically have an ESBL, which will quite happily munch away on aztreonam. The OXA48s can be a little bit tricky. They can actually have susceptibility to the 3rd and 4th generation cephalosporins, but be resistant to carbapenems, and they can often slip into a healthcare system based on the susceptibility to 3rd generation cephalosporin. Before you know it, the isolate gets a porin mutation, and then you've got a horrible outbreak of a truly overt multi-drug resistant organism because of that. So let's look at our traditional, newer agents. So we've got our ceftolozane tazobactam, ceftavazi, meropenem vabibactam, imipenem relebactam, and ceftiricol. You can see just by knowing what gene or protein that you're dealing with in the isolate without actually getting susceptibility to the given agent, you can infer susceptibility or resistance. So we can see if we detect KPC, we're going to be susceptible to all these agents, 9 times out of 10. Class B, you can see you're likely only going to be susceptible to ceftiricol. And then with class D, you can see you're only going to really be susceptible to ceftazidime-avibactam and ceftiricol. So this highlights the power of using these targeted molecular assays for detecting carbapenemase genes. So what about these huge panels that I talked about? Well, at the start. So here's an example of an AMR gene panel, which is developed by Upgen, that detects close to 50 different resistance genes and mutations. And I can tell you it was pretty tiresome to put this table together. But you can see here are the different classes of antimicrobials, and you can see here are the resistance genes that are detected. The one thing I will say, I'm not sure how I would use this clinically yet, because I'm not smart enough to kind of distill this into a report for my physicians. But I'm thinking about it. You know, how will I ever be able to use a panel like this for clinical treatment? Because there's so many genes that kind of throw me and might throw a physician as well. But there is an assay out there that can be used to for bacterial isolates, or also for urine. It semi-quantitates 5 different pathogens, and it has a really quick turnaround time of 3 hours. But I think it does require a little bit of front-end work. But how can these kind of large resistance panels actually be used for the better good of infectious diseases in clin micro. Well, there was a really cool study that I liked that came out a couple of years ago that wanted to understand the prevalence of carbapenem-resistant Enterobacteriales at a time when it was called Enterobacteriaceae in DC. And they rationale correctly that understanding the distribution of CRE colonization is a key component of actually controlling CRE transmission and dynamics within a given area. So what they did is they obtained rectal swabs from close to 1,000 patients that were in 16 different healthcare facilities in DC. And ultimately they showed, just as a point prevalence, there was a point prevalence of about 5.2% CRE in DC. And this serves as a kind of starting point where they can really start to understand what's going on with DC. So do more of these kind of surveillance studies. They can understand, are CRE tracking between particular healthcare centers? Is there an increase in a particular type of CRE? Remember, I showed you, you can actually define what gene is present or many genes, and you can look at many other things other than CRE. You can look at polymyxin resistance. You can look at aminoglycoside resistance, fluoroquinolone resistance. So New York State, which I thought did something that I thought was very cool and very forward thinking, they are working within the state, and it's in this kind of pilot area, to actually develop something like these AMR panels to actually understand transmission of multi-drug resistance within the state. So they're evaluating these surveillance tools. So remember, these panels have to come with some pretty neat informatics that allow the data to be collated and analyzed and understand hotspots and outbreaks. So they're evaluating in New York State surveillance tools with these kind of medium level AMR panels to understand multi-drug resistance dynamics. And ultimately the goal is to have surveillance and control of antimicrobial resistance organisms within the state. I don't think we'll ever have control. We're arrogant to think we've got control of these beasts, but we'll have a damn good shot at doing that. So in my opinion, these panels are very useful for surveillance. And in my opinion, they offer a kind of compromise between whole genome sequencing and those kind of limited panels like the CARBAR panel that I told you about for detecting antimicrobial resistance. Whole genome sequencing is kind of the purview of state labs and public health labs due to the sophistication that is required, although there are clinical labs that are bringing it in. But the AMR panels can actually be used in all labs. These limited AMR panels that I just described can be implemented in any lab. So they offer a kind of nice compromise between practicality and also information that they offer. Another thing I really want to stress is that when you develop these panels, and I think I had before, you really do want to indicate what's been detected, make it very clear to your physicians, what therapy you might recommend for the detected marker, an organism, and also some other comments that you might want to direct your physicians to. So this is just the example that we use for the BCID panel when we detect Staph aureus that's got MEK-A. We say clearly what it is. We indicate what therapy could be used or should be used, and then we indicate what additional steps that the physician should be taking. I think that if you're going to use these panels, particularly from clinical specimens, you should absolutely be providing interpretive comments and information to your dogs. I want to talk about a checklist requirement that kind of blew my mind when it came out. And this was a requirement that basically stated, I'll read it to you if you don't mind. I told you that my superpower was boring people. I'm just about to do that. Basically, there was this checklist requirement that came out and said, direct testing for organism identification and susceptibility phase 2. If organism identification and/or antimicrobial susceptibility testing, phenotypic or genotypic, is performed directly from patient specimens, as applicable, or directly from positive blood culture broths, the specimen blood culture is inoculated onto solid media to assess for purity, and the growth examined for consistency with the ID and AST. Essentially, it was trying to get at the point where we had to try and link up the resistance determinant with the organism that was detected, that was also detected. Think about a scenario where if you've got a polymicrobic blood culture, you've got an E. coli, and you've got a Klebsiella, and you've detected that CTXM gene. That CTXM gene could be associated with either one of the two. And it makes sense that you have to actually, I've got the wrong checklist requirement here, haven't I? I have. I've got the cap list. Sorry, I should. Oh wow, what a moment. The checklist requirement I'm talking to is 21855, which basically states that you have to be able to link up the gene that was detected in the clinical specimen with the organism that was detected. This makes perfect sense. Sorry, guys, got you all scared there. You thought I'd gone crazy. But that's so that checklist requirement 21855 is actually pretty tricky, right? You have to be able to associate the resistance marker to the organism that you detect in the specimen. Not as easy as you think it is, and could cause a lot of kind of work through and a lot of heartache for labs. So recently, CAP removed this checklist, which is resistance terminants and phenotypic AST, where you've got to link up the two in June 2020. So we don't have to worry about this anymore. But it's good practice to be able to link up resistance markers with organisms once you have recovered them in culture. Sorry about that slip, guys. The other thing I'll state, and we're almost near the end, guys, is that it's good practice to get in the habit of resolving discrepancies between genotypic and phenotypic results. Just for the reason I said, one of them is if you detect MEK-A and Staph aureus, but you only recover MSSA. That should prompt some type of investigation and how you handle things. And the CLSI, a society near and dear to me, does have these this appendix H, which allows you or provides suggestions on how to resolve discrepant results between genotypic and phenotypic results for Staph aureus, oxacillin, Enterococcus, vancomycin, Enterobacterales, and fourth generation cephalosporins, and Enterobacterales and carbapenems. In addition, a really nice paper that was just published by Rebecca Yee, an up-and-coming superstar microfellow, working with Jandine Bard and Trish Simner, basically put out a paper saying, how do you resolve these discrepancies? They have a number of different suggestions on how you do that. They have flowcharts, look at the culture growth, look at your instrumentation, look at the reporting, and then they very nicely provide these tables. So let's focus on the gram-positive. Enterococcus, what do you detect, not detect? It's VAN A, VAN B. What are the potential discordant phenotypes and how you should handle them? Similarly, for Staph aureus, what are the potential situations that might arise and how do you handle them? How do you report them? And again, for our ESBLs and our carbapenemases, what are the potential issues and how do you handle them and how do you report? So what are the considerations for using these targeted panels? So the advantages are that they detect genes encoding the exact AMR mechanism, so you know exactly what you're dealing with. They're rapid setup, and typically the results are rapid. They can use a range of clinical input material, clinical specimens or bacterial isolates. They're agnostic to purity. You can use them on a clinical specimen. That's one of their advantages. And you can detect multiple markers simultaneously, and also organisms, as I've shown with those panels. The disadvantages are significant too, and everyone should be aware of the pros and cons of everything that they bring up in their labs. So the disadvantages are they can fail to detect new, uncommon, or novel genes or gene variants that are not included in their panel. Their analytical specificity is both their strength, but also their weakness. If anything changes, as we know with COVID, things are changing, and so assays are, not detecting certain variants. The same is true for these antimicrobial resistance panels. The presence of a gene is not always associated with resistance. You could overcall resistance and take something off therapy. It's not easy to link AMR resistance or the determinant to a particular organism using these methods. Again, that was a CAP checklist requirement to do that, and it was ultimately removed based on a number of folks who had actually talked to CAP about it. And they're expensive. Again, we are still conditioned as microbiologists to think shrewdly and frugally, but we shouldn't, but we are. And they are expensive compared to culture-based methodology. So what do I really think is needed? I think that all labs, this is bold for me to say, this is just my opinion, and maybe I hope it doesn't come across poorly, but I do think that all labs should have the ability to detect or use a rapid diagnostic for testing positive blood culture broths. And there should be an antimicrobial stewardship involvement with the results that are generated. Again, this is a recommendation, albeit weak, with moderate quality evidence put forth by the IDSA. So there is substance to it. It can be used to help persuade administrators to help us if we need these things. There should be therapeutic recommendations posted with the organism, or at least readily accessible to the provider, the patient-facing provider, that tells them how to handle therapeutically the detected organism and resistance determinants. Got to be easy, no more than 3 clicks, otherwise people freak out. We should have molecular detection of MEK-A for surveillance therapy. I think that should be a possibility. I think most labs should have that in place. I think that's important. And often these assays can be repurposed for even therapy. In our institution, someone going into the ICU will get an MRSA screen because it's shown that if they do develop pneumonia, it's likely to be staph, that if they were colonized with an MRSA, it's likely to be that MRSA that's causing pneumonia, and they won't use an anti-staphylococcal penicillin or cefazolin. And conversely, if they were colonized with MSSA and they develop pneumonia, it's likely to be MSSA, if it is recovered, and that they can use an anti-staphylococcal penicillin or cefazolin. I think in areas where there are high levels of CRE prevalence, there should be molecular methods to detect carbapenemase producing enterobacterales. And there are now immunoassays out there that are very, very good, that can do this at the bench in 15 minutes. You don't need a whole molecular setup to do that, like the CARBA-R that I discussed. And then finally, what is really, really needed is close collaboration between clin micro and infectious diseases. And I started off with the importance of antimicrobial resistance, the importance of antimicrobials. And again, I think I've shared this with you all before, but it's worth another showing. This is a statue outside of the bull ring in Madrid. And again, I am no way celebrating bullfighting. I could never watch it. I'm a squeamish human, and it seems a little bit rough to me. Of a statue of Alexander Fleming, who's being saluted by a matador. And why the heck would that even be there? Well, before the advent of penicillin, matadors would get jabbed by the horns of bulls, as they jolly well should be if they're trying to stick them with a sword, and would often die of horrific infections as a result of being jabbed by or engorged by the bull's horns. And so when penicillin came out, a lot of matadors lived because they could treat the infections. Something I found out relatively recently, that I heard that a lot of bull rings actually have operating theaters actually there to handle some of these horrific things that can happen to matadors. Kind of strange. But I also like to highlight the folks that did all the hard work, right? We've got here Alexander Fleming, we always hear about, but Ernst Heim and Howard Florey, who in the Second World War really took on the challenge of getting penicillin and using it therapeutically. Incredible. The 3 of them won the Nobel Prize together. Poor old Norman Heatley, who worked with Howard Florey and Ernst Heim, and actually came over to the US during the Second World War and serendipitously found that one strain that was able to produce a lot of penicillin. He was the fermentation chemist who actually worked out a back titration method to extract more penicillin from the fermentation cultures, significantly more. But because you can only have 3 in a Nobel Prize, poor old Norman missed out. And I will finish here and acknowledge you. I want to acknowledge all of you at JAM. And finally, I've got it right. Steve Jenkins, every time he sees me present, I put jams. He's like, no, it's jam. And I'm like, I can't get my head around that, but I think it's jam. Tell me if I'm wrong. So I want to thank you all. You've been wonderful colleagues over the years, and I love chatting with you. Just a wonderful group of people. I want to thank a number of people who were kind of inspiration for this slide deck, and really want to thank my lab again. And again, thank you all for everything you're doing in COVID. You know, idiots like me who sit in my office and walk around and look at plates. I'm not really doing too much, but everyone who's working on the bench, those gruesome hours, it's all you, and you really are heroes. And I'll stop. I hope I didn't waffle too much.